Good afternoon, everyone, and welcome to our scientific webinar incorporating SNP analysis in the PGT workflow, clinical and research perspectives. For those who doesn't know me, my name is Nasser Al-Asmar, and I am Embryo Product Director and Senior Scientific Advisor in iGenomics, part of Vitrolife Group, in Valencia, Spain. And first of all, I would like to say hi to all of attendees who are joining us today from Europe, Middle East, India, and Africa mainly, but also from Asia and the Americas. A warm welcome to everyone and thank you for joining us. Today's agenda will start with an introduction regarding SMILE PGT-A Plus from my colleague and friend, Dr. Laura Girardi, a Genomics EMEA Scientific Support Manager. And after that, we will have also a friend and colleague, Maria Escrivá, that is a senior clinical embryologist at Equipo Juana Crespo Clinic from Valencia, Spain, that will give us some interesting data regarding atypically fertilized zygotes. Last but not least, we will finish with a Q&A session where we will take questions from the audience. And for this purpose, please write your comments and questions in the Q&A box. You don't need to wait until the end of the session to start asking questions. So whenever you're ready, please let us know what you would like to know. So let's start with the introduction. And as I mentioned, our first speaker is Dr. Laura Girardi. Dr. Girardi was trained in molecular biology, and she obtained her PhD in molecular medicine at Padua University in 2020. She has been working in the field of reproductive genetics since 2015, first as a lab manager in iGenomics Italy, responsible for the PGT diagnostic service, and later as an R&D supervisor. Her main research focused on the development and validation of innovative technologies for PGT, together with basic research and clinical studies regarding aneuploidies and mosaicism in trophectoderm biopsies. Currently, she's the head of the scientific support department at iGenomics, where she's focused in supporting clinics and patients adopting reproductive genetic services. Laura, whenever you want, the floor is yours. You are muted, Laura. Now?
Okay, can you see the full screen?
That's perfect. You can go ahead.
Oh, okay, thank you very much,
Thank you.
Nasser, for the kind introduction, and welcome to everyone. Thanks for joining in this webinar. This is my agenda for today. As you can see, I will start with a short introduction and background about aneuploidies and how to detect them. Then I will focus on our smart PGT-A Plus approach with the new features that we have incorporated, and I will have a special focus on ploidy analysis and pronuclear check. Finally, I would like to share with you the benefit of comprehensive SNP genotyping analysis before going into the conclusion. So I hope you are familiar with the term preimplantation genetic testing for aneuploidies. This is usually considered as an embryo selection tool. Basically, we can count the number of chromosomes. We look at all 24 chromosomes, and we can say if the embryo is euploid, so correct number of chromosomes, or if the embryo is aneuploid, so presence of extra or missing chromosomes. Just for you to know, I want to stress the fact that the aneuploidies that we target in PGT-A are the ones that we call uniform meiotically derived aneuploidies, and they mainly occur during female meiosis, which is the main features. These type of aneuploidies are inherited through the gametes, so what we expect is that they come in a constitutional form, meaning all the cells from the embryo will have the same abnormality. And most importantly, when we transfer these embryos, we know that they are associated with very well known unfavorable outcomes, such as implantation failure, miscarriage, and aneuploid conceptions. In terms of technology, how do we detect these uniform Y chromosome aneuploidies? As you can see here at iGenomics, we use a combination of next generation sequencing and artificial intelligence to analyze the genomic data of IVF embryos. As you can read here, we have two main advantages. We have a high level of automatization. First of all, this is extremely important to reduce the manual handling, mixing samples, and decrease all the other associated risks. And secondly, we designed, developed, and validated a proprietary diagnostic algorithm, and this is important to reduce the subjectivity when evaluating the profile. So overall, this combination ensures reproducibility and quality of the results. I also want to stress the importance of validation, not only technical validation, but also analytical validation, for example, with rebiopsies and embryo disaggregation model. So this is just to highlight that the level of technical and analytical validation of our platform, our reporting strategy, our pipeline of analysis for aneuploidies is supported by this and other numerous published evidence. And here, when we report uniform aneuploidies, we have confirmation rates higher than 99% per chromosome in all the other pieces of the same biopsy. But we know also that there are some limitations in PGTA based on copy number analysis, which is extremely accurate in detecting uniform Y chromosome aneuploidies. But for example, as you can see here, cannot distinguish variation in the number of complete chromosome sets. So this is usually due to a normalization procedure in the sequencing read. So when chromosomes are compared between each other, if there is one extra or missing chromosome, we can clearly see it. But if all chromosomes are deleted or duplicated, so same copy number, we cannot detect it. And here you have just a couple of examples. This is, for example, a euploid female profile based on copy number analysis only. But this can be a diploid female, can be haploid, can be triploid, or can even be a mixture of contamination of external and internal DNA, both females. Similar for these other profiles, here as you can see we have an intermediate chromosome copy number for sex chromosomes, but with PGTA only we are not able to say if it is a triploid XSY, if it is mosaicism for sex chromosome, or even a mixture of male and female DNA external and from the embryo. So how to improve the approach and better classify these profiles? Today, next generation sequencing brings new possibility to overcome these technical limitations, basically incorporating single nucleotide genotyping in our PGTA workflow. And this is exactly where we decided to focus and to invest to bring new elements and incorporate new information. One, for example, is ploidy constitution, but we also are able to get new parameters to improve overall accuracy of PGTA, like contamination detection and cohort check that I will show you later. Now, in terms of technology, we have been able to incorporate these SNP genotyping and SNP markers in the same PGTA workflow. It means that starting from the same trifectogen biopsy and whole genome amplification, we can split this VGA product into aliquots. One will go for 24 chromosome aneuploid screening that we currently perform with ion reprosec PGS kit, and the other aliquot will go for target NGS analysis with a custom panel and a bioinformatic pipeline. So just to give you an idea, these SNP markers are able to provide an additional layer of information beyond aneuploidies, and this goes deeper at the DNA sequence level. So on the right, you see just an example of the SNP pattern distribution based on this information that is called B allele frequency. And obviously is different and provide different profiles depending on the genomic constitution of the embryo. And this is useful to improve our approach and brings these new elements of clinical utility. Now, obviously, before incorporating this approach in the clinical practice, we perform an extensive preclinical validation using both cell lines with known karyotype, but most importantly, diploid, triploid, and haploid rebiopsies, meaning that we tested different biopsies from the same embryos previously analyzed with a different technology that was next generation sequencing. And here you have the publication related to this validation procedure. Moreover, very recently, we also had the opportunity to improve our algorithm. So currently we use a sort of double algorithm strategy. And again, we also validated this additional pipeline of analysis. Now, I want to focus more on one of these new elements of clinical utility that we can obtain with SNP analysis, that is ploidy level information. So basically, by incorporating these SNP markers, we can interrogate these markers across the genome, and we can now potentially say if the embryo and distinguish if the embryo is diploid, meaning two sets of chromosomes, one from the sperm and one from the oocyte. Or, for example, if there is a loss of heterozygosity, so we miss one contribution of one genome, the embryo is haploid. Or if finally, if we have this profile where the SNP show an additional genotype, which is compatible with an additional chromosome set, so a triploid profile. And of note, this was something not possible to derive from standard NGS, thus this profile passed undetected. Most importantly, as a diagnostic consideration, I want to mention that both haploid and triploid are compatible with preimplantation embryonic development, so you will have this configuration among your IVF embryos. And most importantly, triploid in particular can be found in 10 to 15 percent of chromosomally abnormal spontaneous miscarriages. And we know that is usually not compatible with development to term. Well, let me show you the results of our experience with ploidy analysis. In the study that you can see here on the left, we presented the clinical application of our integrated PGTA and ploidy strategy. So here I would like to introduce the concept of molecular fertilization check that is able to improve and assist the regular morphological pronuclear check. Indeed, as you can see here, the main aim of this study was to identify the correlation between the morphological pronuclear status, so what usually the embryologist sees, and the genetically determined ploidy status by our ploidy test in preimplantation embryos. As you can see from the study design, we tested a cohort of apparently normally fertilized embryos, so what we usually call 2PN embryos, but we also put and brought some information on the correlation between pronuclear number and genetically determined ploidy status in atypically pronucleated zygotes. So those embryos showing 0PN, 1PN, 2.1, or 3PN. And this is a large data set that we analyze to provide additional useful information for the management of these embryos. So let's start, for example, looking at the data that we obtain in abnormal fertilization. Here you can see, for example, that not all 2PN embryos are actually diploid, but there is around 1% of embryos resulting in triploidy and haploidy. So clearly, the main benefit here for regarding the use of ploidy test is to have the possibility to detect this ploidy abnormal ploidy configuration and to deselect them from the transfer. And as I was saying, is from apparently normally fertilized embryos. On the other side, another application is related to the testing of embryos showing atypical fertilization during prenuclear check, so 0, 1, or more than 2PN embryos. And as you may know, as routine practice, these are usually discarded due to the high risk of abnormal ploidy constitution. But what we found, which is in line with previous studies, is that this standard morphological pronuclear assessment is not always perfectly associated with the genetic ploidy status. Indeed, there are some evidence that when tested, this blastocyst can display a diploid genetic composition, meaning a balanced constitution from the gametes, and can even result in healthy pregnancy. So, which is our experience? And again, to understand if there is a clear benefit in the application of ploidy testing in these embryos, we use the same PGTA and ploidy strategy to test a large cohort of 380 euploid zygotes resulted after fertilization check as abnormal. So first of all, as you can see, as previously reported, this initial classification of abnormal fertilization based exclusively on pronuclear observation does not always predict an abnormal ploidy configuration. What does it mean? It means that not all 0 or 1PN embryos are haploid and not all 3PN embryos are triploid. And if you follow the blue color, here you can see that there is in different proportion, but the possibility to have diploid embryos and save and rescue these embryos from all the atypical PN categories. Obviously, it's interesting also to work with time-lapse. So after obtaining the expected global ploidy results in a typical IVF scenario where both static and time-lapse observation are adopted, here we had the opportunity to work with a subset of embryos where the pronuclear visualization was confirmed by time-lapse. which are the differences between global data. First of all, when confirming the pronuclear status by time-lapse, the accuracy in predicting the correspondent abnormal ploidy status increases from 28 to 80%. It means obviously that we have more haploid from 0 and 1 PN and more triploids from 2.1 and 3 PN because the PN prediction is more accurate. In parallel, the other consequence is that diploid rates obviously decrease, so we have to expect and be crystal clear in say that we have less diploids that we can say from an observation from time lapse. But obviously there are some categories like 1 PN or 2.1 PN where diploid embryos still persist and can be potentially employed for clinical purposes. Now, just to close the chapter of ploidy, this is another interesting finding, again, from our last paper that we published. And this is to support the use of the small panel that we are currently using in next generation sequencing. Here we had the opportunity to perform an inter-platform comparison where we have independent data set of embryos showing a typical pronuclear pattern, analyzed one dataset with SNP array using 300,000 SNPs, and the other dataset using our AmpliSeq target NGS panel with a reduced number of SNPs. And as you can see here, even if you are not able to read, but I can tell you that we obtain consistent results with different datasets, even the number of selected SNPs was lower. Now, in the last part of my talk, I would like to focus on a more comprehensive approach where the use of single nucleotide polymorphism analysis can assist in improving overall the diagnostic workflow. Indeed, on top of aneuploidy and ploidy analysis, we can now incorporate some additional elements, for example, contamination detection and cohort check. And as you can see on the right, this definitely opened the doors for a broader spectrum of indication for our patients, not only advanced maternal age, but also, again, other indication that you can see here on the right. Let me jump more into the details of these additional parameters. First of all, SNP genotyping data can help in detecting contamination. And I would say this is a quite relevant aspect in our routine practice, as contamination can mask the true chromosomal status of the tested embryos, leading to erroneous results and potential misdiagnosis. It means that now, with SNP analysis incorporating our current PGT approach, we can see if there are two genomes, so more than one genome present in the sample. And these results, obviously, might not actually correspond to the embryo, so it's important to detect them. In terms of contamination ability, we validated levels between 20 and 80% of contamination. And during our validation, we have estimated that around 0.5% of the total trifactorial biopsies can be contaminated. I just want to clarify that when we look for contamination, we look for an admixture of different DNA sources, okay? So clearly, when validated, we have to find that and try to mimic in some way the mixture of DNA. We validated in two ways with mixes of different DNAs in the laboratories in different proportion, but we have also validated in silico mixes using the sequencing files. And these are the different levels of contamination that we can now detect with our pipeline. Let me show you just an example of the effect, the impact on contamination on our PGT profiles. This is a profile where you have some intermediate chromosome copy numbers, for example, one intermediate chromosome copy number for monosomy 21, something that would have been reported as mosaic monosomy 21. But in this case, the variation is interpreted in the presence of contamination as a uniform aneuploid. So we have the opportunity to say uniform aneuploid with contamination. Very important. Additionally, the other parameter very, very relevant is related to genetic relatedness between embryos in a cohort, which is the assumption. If all the embryos originate from the same sperm and the same egg source, we expect to have a sort of sibling relationship between each other. And SNP analysis can help in confirming this relationship and also obviously to detect some mismatches. And most importantly, this will be without the use of parental DNA. Now, in terms of potential mismatches or cohort check inconsistencies, there are many hypotheses behind. First of all, we can have a true mismatch. So incorrect embryo biopsy, incorrect tubing, mismatch in the PGT lab. We can have, for example, embryos derived from different donors. So obviously, if the same couples using different donors, the embryos will not share exactly the same genotype in the same genomic profile. But I also want to comment another explanation, which is complete contamination. So with the contamination test or contamination quality control, we are able to see a mixture of DNAs, two different genomes. In this case, with complete contamination, this does not represent the embryo at all because we only have one external source covering the results of the embryo. So in this case, will appear, for example, as a genetic mismatch with all the other embryos. And again, this cohort check support also in the identification of complete contamination. Now, in terms of results and benefit of comprehensive SNP analysis, I'm pleased to show you some very recent results that we had the opportunity to present as a accepted oral communication at the last PGT-IS 2025 Congress by our colleague Lorena Rodrigo. This is an independent data set that we collected after the publication, are around 3,000 embryos with normal fertilization because 2PN were clearly stated and indicated. Here you see the proportion. Around 1.4% are abnormal imploidy, so triploid or haploid. We also detected 0.5% of contamination, and all core check resulted as consistent, so no mismatches. What is really interesting to see here is how these results would have been reported if only PGTA without SNP analysis would have been applied. So among these 1.4% of triploid and haploid, you see that 63% resulted as intermediate chromosome copy number for autosomes. So clearly, we would have reported as mosaic with PGTA only without the use of SNP analysis to say, for example, triploid. So in this sense, SNP analysis can help in having more accurate mosaicism diagnosis. And this is not only for autosome, it's also for sex chromosomes. Indeed, around 70% of this 1.4% abnormality would have resulted as intermediate chromosome copy number for sex chromosomes. So again, improving mosaicism diagnosis. And finally, this is the more tricky and dangerous configuration, I would say, because 20% of this 1.4% resulted as euploid XX. This is a configuration that, is impossible to be reported as abnormal, as mosaic. So without the use of SNP analysis, we would not have the opportunity to report as triploid or haploid, but just as euploid XX. Another set of interesting results that our colleague Lorena Rodrigo presented at PGT-IS is related to the incidence of abnormal ploidy configuration according to PGTA indication. I think is quite relevant because, for example, if you see here, if we compare advanced maternal age patients versus good prognosis patients, we see a significantly increase in the incidence of ploidy abnormalities. We saw also a higher trend for recurrent pregnancy loss male factors versus good prognosis, but is more significant in advanced maternal age. And if we look specifically at triploid configuration, we saw a significant increase in terms of incidence in advanced maternal age versus good prognosis. And again, just a higher trend in repeated recurrent pregnancy loss versus good prognosis. Haploid seems to have a higher trend in recurrent pregnancy loss and male factor, but is not significant. So very, very interesting here is that not only aneuploidies are associated with maternal age, but also triploidy has a strong association as well. And in my opinion, this is an important diagnostic consideration for PGTA because, as we said at the beginning, this is one of the most leading cause of miscarriages. So obviously, every PGT workflow should move forward to incorporate some form of ploidy level to detect and deselect these ploidy abnormalities. So let me jump into the conclusions. I want to remark that the main aim of PGTA is always to detect uniform Y chromosome aneuploidies, no matter if it is with a more accurate and more sensitive technology. But obviously, with more sensitive platforms and the addition of SNP analysis, we can now provide additional levels of information and clinical utility. From our published and new data, we found that around 1.4% of blastocysts derive from normal fertilization, so 2PN, they are triploid or haploid. And on the other side, the other interesting finding and benefit is that abnormal PN configuration, all of them, show some evidence of diploid, so we can use potentially these embryos. We saw also that PGT with ploidy and contamination analysis decreases the risk of misdiagnosis because we can identify now if there is the presence of a non-embryonic DNA, so a contamination, and overall we can improve the diagnosis of mosaicism and embryos with intermediate chromosome copy number on sex chromosomes. Quality control parameters like contamination and cohort check provide additional reassurance to your patients, and as our policy, they are clearly indicated in the report, which can also contain additional support to guide the decision-making process on the use of these embryos. And finally, take-home message, the incorporation of SNP analysis in PGTA results in a more accurate diagnosis with potential improvement of clinical outcomes. So thank you for your attention, and Nasser, now the floor is yours again.
Thank you. Thank you very much, Laura, for this fantastic presentation, as always. You were able to bring it up some really interesting data coming from PGTIS last week, actually. So it's really nice new data. So thank you very much. And I will see you in the Q&A session.
Yes,
Thank you.
thank you.
Perfect. So once again, please write your comments and questions in the Q&A box. So far, I can see only one question. So please don't be shy and go ahead to make your questions. Now let's start with the presentation from our second speaker. Let me introduce you, Maria Escrivá. Maria earned her master's degree in biotechnology of human-assisted reproduction from Ibian University of Valencia in 2013. Following this, she moved to London, where she advanced her career as a clinical embryologist and began developing a parallel interest in research. In 2017, she joined Equipo Juana Crespo Clinic in Valencia, Spain, where she now works as a senior clinical embryologist while actively participating in research studies. Her work primarily focuses on PGT and embryo quality with the aim of continuously improving clinical outcomes. Many of her studies have been presented at major assisted reproduction conferences, including ESHRE, ASRM, and PGTIS. So Maria, whenever you want, the floor is yours.
Thank you. So hello everybody. My name is Maria Escrivá and I'm a clinical embryologist in Juana Crespo in Valencia. First of all, I would like to thank Nasser and Economics for inviting me to share our results using Smart PGT Plus in clinic. So, and today I'm going to talk about two types of 2PN, of 3PN embryos and their genetic outcomes. So as we know, in an IVF lab, fertilization is commonly confirmed around 17 hours after conventional IVF or ICSI, with the appearance of two nuclei and the extraction of the circumpolar body. This is what we call a 2PN embryo, but sometimes we observe a third nucleus, indicating an atypical fertilization. We call to these embryos 3PN embryos, and they are often discarded due to the risk of being triploid or having an extra sem of all chromosomes. But the question is that true? Are all 3PN embryos real triploid embryos? So in 2017, Capalbo and colleagues proposed a new way to look at these 3PN embryos. They divided them into two groups according to the size of the third pronucleus. They only analyzed embryos with two pronuclei of similar size and a third smaller one, believing that this might have a better prognosis. They called to these embryos 2.1 embryos. They even transferred some of these embryos, and as a result, they have two healthy babies. And that finding proved that viable embryos could be potentially rescued from a pool of 3PN embryos. As you know, a couple of years ago, Genomics launched Smart PGT Plus, and as Laura has explained in this such a brilliant way, I won't explain again. But this PGT program has different advantages. Today we'll focus on the predictions using SNP analysis, but I would like to say that from the point of view for embryologists, another advantage is that this is very easy to use. It's the same procedure as a regular PGT biopsy, same tubing, and we don't need parental samples to analyze this. So with the Smart PGT Plus, we can potentially rescue some embryos that otherwise would have been discarded in clinical way in the lab. So having this background about Capalbo and so many others, and with this new technique that we can use in an easy way, we started this study to compare both types of free PNs. We wanted to see if they behave in a similar or in a different way. And the most important, we wanted to quantify the percentage of embryos that we could rescue to see if it's worth to keep and analyze them or not in benefit for patients. We have been running this study for a long time. We presented the preliminary data in April last year, and last week we have been in PGDs presenting the updated version. So here we have our study, and it's a prospective observational study that includes the data of 349 free PN embryos obtained of 317 cycles of 297 patients, who all of them consented, obviously, to be in the study. We included all kinds of treatments, such as ONEX, egg donation program, free-sol patients, PGT patients, all kinds of treatments. And it was performed in only one lab, in our lab in Valencia, in Spain, in Equipo Juana Crespo, from June 2023 until December 2024. During that period, our free PN incidence was 2.0%. So the first thing we did was to define both groups. The first group is the commonly known as the 2.1 group, and in these embryos, the diameter of the smallest pronucleus is less than 70% of the diameter of the largest pronucleus. We draw the line in the 70%. Here we have an example of a 2.1 embryo where we see this small pronucleus there. We know that these 2.1 embryos sometimes are very difficult to see, to spot them, and sometimes the small PN can hide behind the large one. So even if you're focusing at different planes in a conventional microscope, you can see it, but with embryoscope, it's much easier to spot because you will see it eventually. Another thing that is quite common to happen is that sometimes you observe you're doing the first check and you see that you have two regular PNs, but you see something else, and you're not sure if it's the third PN or not. It could be a bug or something, you're not sure. If you're using a conventional microscope or even in embryoscope, but with only a static image, it's difficult to recognize. But with embryoscope, using the video and moving back and forward, it's quite easy to spot, to tell if it's a 2.1 or not. Because if all three PNs are moving in a synchronized way, it's a 2.1 embryo. Otherwise, it's not. And they will fade at the same time eventually. So here we have like an example also where this is the same embryo, and moving back and forward, we see all three moving in this synchronized way as I was telling you. Another advantage of using embryoscope is that sometimes, this is not quite common, but it happens that sometimes while you're doing fertilization check, you observe only two PN, so it's a fertilized embryo. But the day after, on day 2, you check your embryo, and after 17 hours in culture, so after the fertilization check, you observe a third pronucleus. In a conventional incubator, you will miss that, but having the embryoscope, you are able to see it, to spot it, so you can call the patient and explain her and advise her in the best way. Like in this example, where at 17 hours, we see only 2 PN, but after that, now, another one appears. So you know that it's a 2.1. Otherwise, you wouldn't have known it. And the other group is what we call a same-size 3PN. In this group, the diameter of the smallest pronucleus is more than 70% of the diameter of the largest pronucleus. Here we have an example, and these ones are very easy to recognize. Okay, so now we have the results. We'll start with the 2.1 group, okay, and we had 189 embryos. Okay. Out of these 189 embryos, 69 reached good quality blastocyst stage. We define good quality as quality enough to be frozen or transferred. All of them were biopsied, and as a result, we have 64 samples informative for ploidy and uniploidy analysis. Out of the 64, 57 were diploid and 7 were triploid. Most of them were diploid. And out of these 64, 12 were euploid, 3 were mosaic, and 49 were aneuploid. As a result, we had 15 transferable embryos, 12 diploid euploid, and 3 of them were diploid mosaic. On the other hand, we had the results in the same size group, and we had 160 embryos. Out of them, 60 reached good quality blastocyst stage, we biopsied them, and 48 of them were informative for ploidy and aneuploidy analysis. As a result, out of these 48, 2 of them, only 2 were diploid, and most of them, 46 were triploid. 19 were euploid, and 29 were aneuploid. And we had 1 transferable embryo, 1 was diploid, euploid. So now we're going to compare these outcomes. First we'll talk about blastocyst rate. And with blastocyst rate for the 2.1 group was 36.5%, and blastocyst rate in the same size group was 37.5%. So no significant difference were found between both groups, but both blastocyst rates in the 3PNs are lower than the blastocyst rate for the 2 PN embryos, those sibling embryos on those 317 cycles. So the blastocyst formation in a 3PN embryo is lower than expected for a 2PN embryo. We have very interesting results in about ploidy status, as 57 out of 64 were diploid in the 2.1 group, and only 2 were diploid in the same size group. So the diploidy rate were 89.1% in the 2.1 group versus only 4.2% in the same size group. So obviously this show significant differences between them. We can't draw very definitive conclusions about ploidy status of these embryos, as we are very aware that 112 samples are not enough to draw definitive conclusions in a PGT program. But what we can see is that it appears that the same size groups have a similar euploidy rate compared to 2PN populations, and is very age dependent. But in the other part, we see that the euploidy rate in the 2.1 group is lower, despite being younger, and we think that this could be due to the own nature of these embryos. But anyway, we have compared both aneuploid rates, and despite being higher in the 2.1 group, no significant difference was observed in aneuploid rate. Another problem was that the age of our patients was very different, so that's why it's very difficult to draw very definitive conclusions. But the important thing here is that we observe that euploid population exists among all 3PN embryos. And this is the final part, the most important part, where we see the rates of the viable embryos in both groups. And as a result, we had in total 16 viable embryos for patients, 15 in the 2.1 group, and only 1 in the same size group. So as a conclusion, we can say that almost a quarter, 23.4% of the 2.1 biopsied embryos are viable embryos for patients, versus only the 2.1% of the same size biopsied embryos, that only those 2.1% are viable embryos for patients. And that's quite different, with significant different significances, obviously. So the take-home messages are it is very important to differentiate the two types of the free PNs, as this will help us to know the potential of the embryo and advising the patient in the best way. Most of the 2.1 free PNs are diploid, almost 90%, so it's worth to biopsy analyze them using SNP analysis, as it will inform us about the ploidy and the euploid status of the embryo. But we can't forget that despite most of them being diploid, the remaining almost 11% of 2.1 embryos are truly triploids. So we shouldn't transfer them without a previous analysis, as they could result in a triploid pregnancy and a molar pregnancy. Regardless being triploid or diploid, embryos could be euploid, mosaic, or aneuploid. So with PGT-A is needed to check if embryos are suitable for transfer. And as conclusion, 23.4% of the biopsied embryos are viable embryos, increasing the number of viable embryos as a benefit for the patient. I would like to thank the genomics team because of their support and their work, especially Carmen and Lorena, and obviously my colleagues in the lab for making this possible. So thank you very much.
Thank you very much, Maria, for your super nice data and presentation. And again, data coming from last week. So pretty new data for all of us in order to try to apply this kind of tests at the clinical level, right? And I'm going to start with you, actually. One question that I can recall from PDIS as well, that is about clinical data. Question is, from the 16 embryos available for transfer, coming from 3 PN, 2.1 PN, did you have any clinical data? So are you transferring already a few of them, or?
Not yet, because now we are transferring embryos to the patients that were 2PN if they have. Only if there are no embryos available for patients, we will transfer these embryos. So we are waiting now because there's a couple of patients that maybe we'll transfer in few weeks. But for now, we don't have clinical results.
Okay, so because I guess that obviously you are going to be transferring first those euploids with 2PN, and eventually, okay, cool, perfect. Laura, there's a question asking about report. They're asking what kind of support you give to the clinics with a result with contamination or with inconsistent cohort check result. How do you deal this with the clinics, and the clinics what can they do with patients?
Yeah, absolutely. Thanks for this question. Is very interesting indeed. For example, when we talk about contamination, we have to expect that we have some, additional DNA that is in some way covering the results coming from the embryo. So, if it is aneuploid or if it is an intermediate chromosome copy number, we had the opportunity to report it as aneuploid with contamination. But for example, if it is euploid, in this case, if we detect contamination, we also add a sort of comment, a sort of support in the report that is like rebiopsy and retesting. And I think that this procedure of having a second biopsy and retesting can be considered and is acceptable in case of a euploid results, because we want to be really sure that now we are with a second biopsy seeing the real genomic constitution coming from the embryos. Obviously, if the embryo is of appropriate morphology and quality to have a second biopsy. This is more or less what we advise and suggest for contamination. For cohort check, usually we do not report the results. What does it mean? It means that if we see a sort of inconsistency, which could be compatible with a mismatch, but in any case, we don't know the source or the origin of this inconsistency. First of all, we don't report the results, so we don't report or say which is the results. And secondly, we don't release immediately the report with cohort inconsistency reported, but we call the clinic and we try to find at which stage this mismatch happens. So first of all, obviously we look and we go back in our PGT workflow to see if there are some internal mismatches, because you know that we analyze more embryos at the same time, so we can easily see if there is a mismatch among the same embryos in the same analytical session. And then obviously we call the clinic, try to identify the potential mismatch. Only in the case there is no potential explanation for this mismatch, so the most reasonable explanation is a true mismatch, then we are going to report. But just to give you an idea, so far we only had one or a couple of cases, because the other cases that we reported as cohort inconsistencies were, for example, using different donors or different oocytes for the same couple. So actually was not a real mismatch. So we had the opportunity to release then the correct report without, I mean, giving any sort of alarm to the clinic and to the patient. This is more or less our strategy.
Perfect. Thank you very much, Laura, for sharing. And I do think that this is really important. You show the data about contamination that right now it's around zero 0.5. I think in our validation we estimated less than zero 0.8, but it's still like a number that is there. And also for cohort inconsistency, it's very low, as you mentioned. IVF labs, genetic labs, we work really good on safety. But
Yeah.
last week, we observed, well, we had a case in Australia that unfortunately a baby was born and was not from the parents. So I do think that putting extra tools for our peace of mind, both in IVF labs and the genetics labs, and looking for the patients, I do think that is going to improve our results. And Maria, this is a question that is related with this. You presented data on 2.1 and 3 p.m. blastocysts, but do you think that it's also useful to have contamination and cohort check in a PGTA result?
Yeah, definitely. As what you have said about that case in Australia, that concerns a lot to patients. Even if we have witnesses and everything, patients are always concerned about having this issue. So the better evidence we can give them, the better for them. So this is great for them. And about contamination, possibility is always there. So checking this, so you can check it, you can tell us, okay, this has happened. We can rebiopsy the embryo. That's great for patients also. Otherwise, it will be reported on as a false positive or negative. So that's great. Yeah.
Yeah, and Laura presented actually a few examples and the profiles that we will see if we don't just make the proper call, right? We will be like maybe over calling or under calling if we don't check for that.
Yeah, exactly. And this is both for triploid and haploid configuration and also for contamination, because in some way the copy number analysis only is telling us something that is different from the real genomic constitution. So as you were saying, maybe the risk here is overcalling mosaicism, and we know now with the new evidence that we have that any sort of low-level mosaic can now, I mean, is now accepted and acceptable to be used for transfer. So the risk is to transfer maybe a uniform aneuploid that was reported as just a mosaic due to this intermediate chromosome copy number. So yes, it's very interesting, because if you consider the percentage of contamination, 0.5%, and abnormal ploidy constitution, 1.5%, is more than 2% of cases that we can better classify and better diagnose, avoiding this misinterpretation, as Maria was saying, false positive or false negatives, for sure.
And that's important. And Maria, I have a question also that you already addressed. Actually, I saw you in your final conclusions. So I do think that it's important to remark that you said most of 2.1 p.m. were diploid, but still some of them were triploid. So I guess it
Around
makes sense to
11%.
still around 11.
Around
So
11% of them are triple, yeah.
okay, so the question was like, so I guess it makes sense to still analyzing those 2.1 p.m., right? And this is something that you put
Definitely.
on your conclusions.
Mhm. Not only to avoid negative miscarriages and worst case scenario, a molar or a colonic pregnancy.
Yeah, and actually, you mentioned that molar pregnancy, that before, when a patient had a molar pregnancy, the clinician or the medical team used to ask us if we can do something for the next cycle, and we couldn't. But now,
Mhm.
most of those molar pregnancies that come from triploids, maybe we can do something else and to analyze that. Perfect, thank you. And this is a question for both. They said they're asking about the indication. What would be it's there some indications related with this kind of test? Can we make it broader? Like, okay, we are offering PGTA. Now that we have a better tool, should we move all patients to the PGTA plus? Laura, Maria.
Yes, absolutely. I mean, in my opinion, the indication are first of all the same as PGTA indication, because as I was trying to mention in my presentation, the main aim of this test still remain the selection of uniform meiotically derived aneuploidies. So all the indication related to aneuploidies. What we saw, for example, the additional indication related to the potential consequences of transfer of triploid and haploid, like for example, miscarriages after PGTA. So maybe detecting triploidy now can explain, this percentage or part of this percentage of embryos resulted as euploid in PGTA but ended in a miscarriage or implantation failure. So this is another indication. And as I was trying to show related to maternal age, this is a common indication. So if we have patient with very advanced maternal age, the risk of aneuploidy is higher, but also the risk of triploidy is higher. So for sure, the one that you were mentioning, molar pregnancy, miscarriages, advanced maternal age still remain good indications for this test. Absolutely.
Yeah, and as you both know, in PGDs last week, everything was about the SNPs, because we have seen not only atypically fertilized, can be haploid or triploid, even 2PN embryos, around 2%, Laura and Roena said, are
Yes.
are abnormal embryos. So if we move with this new technology, we will be able to get more information for the patient for their safety. And the same with contamination is low, but it's still there. It can be there. And in the worst scenario is about the mismatch of the samples. So will happen only once, but if that happens, that's a terrible mistake. So if we can avoid that, much better for the patients always.
Yeah, agree absolutely, especially on the last point, because if we talk about indication, because there is a clear, benefit in excluding some categories like triploidy, totally agree, but also why not proposing something that is in general more accurate in calling mosaicism, in reducing mismatches, in reducing misdiagnosis. So I think this is for all patients. We should give them the option to have a PGTA assay with higher accuracy overall, I would say.
Yeah, we always say that less than 0.1, it's super low rate, but if it's yours, it's 100%, right? So it's complete disaster. Okay, we must finish. We only have the last question before closing the session. I think this is related with PGTA in general. It's thank you for amazing lecture. Just one question: have male factors also been associated with aneuploidy rates detected by PGTA?
Male factor, I would say that is more maybe an indication for PGTA plus in general, because for example, if you have like a high concentration of diploid sperm, for example, this could be an indication for testing for ploidy. This is my recommendation here, for example.
Yeah,
It's
because
quite controversial. It's not that clear at advanced maternal age, but something is there for sure. Yeah.
yeah, we don't know that the lower sperm concentration, the higher rates of abnormal fish on sperm. And as you, Laura, mentioned, if we have higher concentration of diploid sperms, then when fertilize an oocyte that is haploid, we will become triploid blastocyst. So then
Yeah,
it would be
more
worth it
frequently.
to make this kind of analysis. Okay, cool. So, thank you very much for your explanations. Fantastic data, super new data, and this is something that is encouraging us to do more molecular pronucleus analysis, as Laura was explaining at the beginning. Because Maria, with these kind of things applying to the clinic, I do think that the rest of our colleagues will be like for sure eager to try to start testing for their patients. And thank you to both of you. I don't know if you want to say a final statement, Laura. You want to say something just to close the session?
Yeah, just want to say thank you to everyone, and as I've always been enthusiastic about new developments and new things. So this technological evolution is for sure bringing these new elements, which are not only like something that we want to report just because it's a trend, but because they have a real clinical utility, and we have been able to prove it. So hope to have the opportunity to collaborate with all of you in the future.
Yes, I would like
Thank
to
you
say
very
that
much.
I would like to say that we have seen 24.3%, I think, that we can rescue from these biopsied embryos. It's a high number, especially with on patients with lower prognosis, with low X, and that can make a difference for them.
Yeah,
So
absolutely,
it's
because
quite
we always
worth.
think,
Yeah.
yeah, that we will have diploid with 2PN, that obviously we will prioritize them for transfer, but then
Mhm.
we have those patients, last embryo, they have to move into egg donation program, but now we could be rescuing this percentage that you mentioned, Maria.
Yeah.
So
Or
thank
even in
you very
future
much.
for a sibling. Yeah.
Absolutely. So thank you very much again to all the attendees, to our speakers for being here today, and hopefully we will see each other in a new webinar. Thank you all. Bye-bye.
Thank you.
Thank
Bye.
you. Bye bye.